Welcome everyone, it's Dr. Lindner. In this video lecture, we are going to be talking about the classification of tissues. And when we talk about tissues, we're going to be focusing on four different types of tissues. Now remember, from a previous lecture, we have chemicals or chemistry, whether it's carbohydrates, proteins, and fats, or carbon, hydrogen, oxygen, and nitrogens, where you have chemistry that comes together to make cells, and then you get a group of cells that come together that have similar structure and function, and that's going to create tissues. And we have four primary types of tissues: epithelial tissue, connective tissue, neural tissue, and muscle tissue. In the first part of this lecture, I'm going to focus on a little introduction, and then we're going to get right into epithelium. And then we'll take a pause, we'll take a break, see if there's any questions, then we'll come back and then we'll do connective tissue. And then we'll take a little break and we'll get into neural tissue and muscle. Of the four, these two tend to be a little bit more involved, not difficult. It's just there's more layers to it. And neural and muscle is a lot easier to learn and understand. But let's see if I could just lay down a little basic concept differentiating epithelial from connective tissue. And you could just mute your mics, that would be great. Hearing some noise in the background. When you look at epithelial tissue is made up of a bunch of cells that are closely packed together, like we see here. This would be under a microscope. We have a nuclei or nucleus for each cell. And notice they're closely packed together. And this is just like one cell layer thick, but I could always add another cell layer to these. And again, you see how closely packed these cells are to one another under a microscope. And this is one of the characteristics to epithelial tissue, is that the cells are closely packed together with very little extracellular matrix. ECM stands for extracellular matrix. Extracellular means outside of the cell. It's the substance that's found between the cells. So with epithelium, the cells are closely packed together. They have very little extracellular matrix, and that's very different than connective tissue, where connective tissue we can have, let's say, a red blood cell here, then maybe we have a type of white blood cell with a different shaped nucleus here, and then we have maybe another type of white blood cell with a different type of nucleus here. Notice the cells are not closely packed together, and there's lots of extracellular matrix or substance between the cells. That's the difference between connective tissue and epithelial tissue under a microscope, okay? Just a little foundational difference between the two. So connective tissue has lots of extracellular matrix, and if this was blood, the extracellular matrix in blood is actually called plasma. The plasma is the substance between the cells. So when we talk about tissues, tissues do have unique structure, whether it's structure, patterns, or form. That is the same thing as form and structure, and that kind of determines its function. And tissues are organized into organs, right? Cells make tissues, tissues make organs, like the heart and the kidneys and the lungs and the stomach. And those two, the arrangement of tissues in an organ, they're going to determine the structure and function of the organs. But when we're studying the tissues, we call that histology. Remember, when we study cells, it's cytology. when we study tissues, it's histology. Now, the first type of tissue we're going to focus on is epithelial tissue. Now, in terms of function and location, generally speaking, epithelia or epithelium, they're both interchangeable terms. Epithelia or epithelium is the type of tissue that lines, it's a lining and a covering type of tissue. So when we think of covering, I think of parts of the skin like the epidermis, the external layer of the skin, right? It covers the external surface of the body. That's a covering. When we talk about a lining, epithelium can line body cavities. Remember that from a previous lecture? We talked about body cavities like the abdominal cavity, the cardiac cavity, the peritoneal cavity, and it also can be found in tubules. Now, tubules, you see the word tube. So anywhere we have something that looks like a hose within the body, a hose-like structure, that's a tube. Whether this is a blood vessel, whether it's a blood vessel, whether the tubing is part of the gastrointestinal tract, the GI tract, whether it's part of the reproductive tract, the respiratory tract, right? The digestive tract, the esophagus is a tube, the small intestine is a tube, the large intestine is a tube, the ureters are a tube, the urethra is a tube. Okay, the bronchi, they're tubes, they're just smaller tubes. But everywhere we have tubes, we're going to have epithelium. And also, we'll want to throw glands into that category. Glands are made up of epithelium. Now, when we look at epithelium, or let's say you're looking at a slide under a microscope, and you're asked to identify the type of tissue, we have to ask ourselves a few questions. Now, remember, when we spoke about epithelium and connective, one of the questions we have to ask ourselves is, hey, do we see a bunch of cells together, and are they closely packed together? If they are, it's going to be a type of epithelium. If it's not, perhaps it's connective tissue. So one of the questions, the first question we're going to ask ourselves is, are the cells closely packed together or not? The second question we're going to ask ourselves is, hey, how many layers do we see? Do we see one layer of cells, or do we see more than one layer of cells? And if we see one layer, it's referred to as simple epithelium. And if we see multiple layers or more than one layer, then we're going to refer to it as stratified epithelium. And the layer that we're looking at is the layer at top. It's the apical layer, the layer at the apex or at the top surface. So that's the second question. And then the third question we're going to ask ourselves is, what's the shape of the cell so that we can name it? We already said if it's one cell layer thick, it's simple. And if it's more than one cell layer thick, we call stratified. So if we see something that's irregular shape like this, we're going to call it a squamous cell. And then if we see something that looks like a box or a cube, we're going to call it a cuboidal cell. And then if we see something that looks like this, like a tall column or a building, we call it a columnar cell. So we have squamous cells, cuboidal cells, and columnar cells. So when we look here, we see that these cells are these flat, irregular shaped cells, and we see that it is one cell layer thick. So because it's one cell layer, it's simple. Because they're flattened, it's squamous. So we call it simple squamous epithelium. To the right, we see that it's multiple layers, right? We have 1, 2, 3, at least 4, it looks like 5 layers of cells stacked. They're closely packed together, so we know it's epithelium. And we know that because it's multiple layers, it's stratified. Because they're flattened, it's squamous, so stratified squamous epithelium. Below it, we see these cubes. So we know that they're cuboidal. And then is it one cell layer or more than one cell layer? It's only one, so it's simple cuboidal epithelium. To the right of that, we have these cubes, and they are more than one cell layer thick. So we know that it's stratified. We know that they're square shaped, so it's cuboidal. So we have stratified cuboidal epithelium. Then down below, we see these columns, and it's one cell layer. So we call it simple columnar epithelium. And then to the right of it, we see that it's more than one cell layer, so it's stratified columnar epithelium. Now, to the right of that, now, one thing I want you to pay attention to, when you look at columnar here to the left, we see how all these nuclei are at the same level. So we know it's one level, it's one layer. But here, these are at one level. This is at another level, all equally. So now we have stratified columnar. But to the right, it looks like, okay, nuclei is at this level. Then we have nuclei at this level. Then we have nuclei at this level and nuclei at this level. So it looks like it's stratified, but it really isn't, right? It's just that the nuclei are scattered at multiple uneven levels. So they call it pseudo stratified. And the word pseudo means false, right? So it's pseudo stratified. It looks like it's multiple layers, but it's really not. So we're going to give this the term pseudo stratified columnar epithelium. But notice that there's something here on the top. These are called cilia. And there's cilia that we find on cells. And here we see them at these cells. And these beat rhythmically. They beat rhythmically, and they're designed to kind of move mucus along the surface. So let me see if I can erase this for a second and draw something here for you. So here, let's see if I could draw this differently. Let's see if I could draw this. So here is, we're going to say this is the trachea. All right, here's the windpipe. So you're breathing air that's going to come in here. And this, again, is a crude drawing. And then let's say here, these are going to be the columnar cells. Okay, let's draw a bunch of them here. So these are going to be columnar cells. We're going to see them on both sides here, like this. You get the point, right? So we have all these columnar cells, and let's pop a quick nucleus in these. We'll put them at different levels so that we'll say it's pseudo-stratified. And wherever we have pseudo-stratified, there's going to be a unique feature that I was talking about before. Let's make this blue. There's going to be these cilia that are on the cell surface on both sides here, okay? And these cilia have a very important function. They're going to be beating rhythmically. They're going to be beating up in this direction, upward, from inferior to superior. Now, there's also, let's make it green here. There's going to be these scattered cells. I'm doing it green with a G because these are called goblet cells with a G. And these goblet cells are going to secrete a substance called mucus. And it's going to push the mucus to the cell surface, and it's going to stick right here to the cilia. Now remember, the cilia are beating upward in this direction. You're breathing in air this direction. It's got dust particles, debris. Maybe the person's a smoker and they're breathing in smoke and debris and carcinogens and chemicals. So the air is coming in this way, and you have the cilia beating upward that has this mucus sticking to it, and it's going to act like a glue that traps any foreign matter and debris against it. so that it doesn't make it, all the debris doesn't make it down into these primary bronchi that eventually will lead into both of your lungs. Okay? So it traps it, and then all you have to do is like this, and then you can force all the gunk outward. In smokers, however, smokers actually paralyze these cilia. They paralyze the cilia or burn them off. And when these cilia get burned off, these pseudostratified ciliated columnar epithelial tissue, they change their shape. They go from columnar cells and they go to these squamous cells. And now, whenever you have a cell that started out as, let's say, columnar or pseudostratified ciliated columnar, and they evolve and they turn into squamous cells, we call that metaplasia. Metaplasia, women are quite familiar with this because when women go to the gynecologist and they do a pap smear, the pap smear really is a histological biopsy, right? They're sloughing off tissue from the cervix and the vaginal canal, and they're looking at the shape of the cells. And they'll call it cervical dysplasia. Dysplasia means they're altered in shape. It's the same as metaplasia. And metaplasia is typically precancerous. That's why they'll often do pap smears twice a year on women, especially when they see cervical dysplasia. They want to make sure that they have not turned into cancerous cells. Okay, so metaplasia, in this case with smokers, oftentimes the pseudostratified ciliated columnar epithelium, they change their shape and they become squamous cells. And you've heard of squamous cell carcinoma. That's throat cancer. Squamous cell carcinoma happens in smokers all the time. Okay, so there's a little bit of a practical understanding of why it's important to understand what these tissues are and how they can change and how they can modify. And the body's always trying to adapt. When things are toxic to the body, cells will die, and then metaplasia kicks in, which is a maladaptive process. And metaplasia is typically a precancerous type of condition. So again, when we have one cell layer thick right here, it's simple squamous because these cells are these flat, irregular shaped cells. So that's simple squamous. Once we see that it's multiple layers of flat, then it's stratified squamous. Then we have simple cuboidal because it's one layer of cube cells. And then we have stratified cuboidal, which is one, two layers of the cube cells. Notice here, these nuclei are at one level, and these are column shaped. So they're simple columnar. And then down below, these are columnar, but we can see the nuclei are scattered at multiple levels. So we use the word pseudo stratified columnar. And typically, wherever we have pseudo stratified, remember we're going to have the cilia associated with them. And these cilia, or ciliated or pseudo stratified ciliated columnar epithelium, are typically located in the respiratory tract. They're also found in the reproductive tract, the reproductive tract. Okay, so here's a new one, this one called transitional. Transitional, it sounds like the cells are in transition. They can vary in shape and size. Think of the urinary bladder, and I'm going to make an analogy that the urinary bladder is very similar to a balloon. A balloon can be distended or it can be relaxed. You can blow up the balloon and it can get larger, or you can let the air out. A urinary bladder can be distended, filled with urine, or it could be relaxed after urinate. It goes back to its normal size. So if you were to, let's say, have a balloon, let's say here's a balloon, and let's say you were to put a box, like draw with a marker, a box like this. But then you push air in here and you inflate the balloon. What happens to this box? Doesn't it alter its shape and change in shape and size? Maybe it's going to look like a circle. Maybe it'll look oval. It alters its shape and size. So transitional, we're always going to find that somewhere in the urinary tract, whether it's the urinary bladder, the ureters, the urethra, because the urinary bladder can be distended or it can be relaxed. So what are some of the functions? I've seen this show up on some of the McGraw-Hill Connect lab quizzes. It's kind of like a drag and drop. So what type of epithelium can function in diffusion, filtration, secretion, and protection? And that's going to be simple squamous. So when you have the trachea, and then you're going to have the bronchi, and then eventually you're going to have the lungs, right? The alveoli of the lungs, the deepest part of the lungs. So somewhere in here, we're going to have these grape-like structures called alveoli. And in the alveoli, I'm going to just draw something here. It looks like a flat, irregular shaped cell. That's a squamous cell. It's one cell layer thick, so it's very easy for oxygen to pass through a layer that's one cell layer thick, and then it's easy for CO2 to pass in the other direction, right? So it's good for diffusion. It's allowing the gases to diffuse if it's one cell layer thick and they're very thin, flat, irregular shaped cells. That's simple squamous. But if it's stratified squamous, if you have many layers of these, then it's really not the best for diffusion, right? Things aren't going to pass through, cells that are 25 to 30 layers thick. But if something is 25 to 30 layers of stratified cells, like in the skin, well, that's great for protection, to protect the underlying layers. So stratified squamous, we'll see that in the skin, especially where we have the epidermis. Epidermis, okay? Simple cuboidal, these are going to be found where the kidneys are located. When you get into anatomy and physiology too, you're going to be learning about a structure called the nephron, and kidneys have millions of nephrons, which is the functional unit of the kidney. You're going to be learning about something called a proximal convoluted tubule, and if there's a proximal convoluted tubule, there'll be a distal convoluted tubule. Where we have the proximal convoluted tubules and the distal convoluted tubules is where we have simple cuboidal. And at the level of the kidneys, it's good for absorption, secretion, and protection. The pseudostratified columnar, this is what I was drawing earlier with the windpipe, where we called it the pseudostratified ciliated columnar. We had the cilia beating rhythmically, and it had those goblet cells, which are going to push mucus across that apical surface. Okay. And then we have transitional, which we're going to find in, let's say, the urinary bladder, where it could be distended or even relaxed. Okay. It can alter in shape, so the cells are constantly in transition. So we know that the cilia are those long, slender extensions. They're going to move secretions and fluid across the cell surface, even mucus, right? Even mucus. And then smaller cilia are called microvilli, and these are smaller projections. And these are designed to increase absorption or secretion, but mostly absorption. So we're going to see microvilli. Let's say here's the small intestine, which is a tubule, and we're going to have, let's say, columnar cells here again, but this time we're going to find microscopic cilia. They're much, much smaller. These are called microvilli, and microvilli are going to act like a sponge. It's for absorption. It's to pull in nutrients so that the microvilli absorb those nutrients, eventually will send them into your blood vessels to absorb all those nutrients, all of the carbohydrates and proteins and fats and vitamins and minerals and sugars, and transport them to other cells and tissues of the body. So we need those microvilli. When these microvilli here are damaged, let's say in celiac disease, people that abuse alcohol, eat too much fried foods, excessive carbohydrates and high sugary foods, overuse and abuse antibiotics, they damage the microvilli, and we can create these malabsorption diseases, malabsorption, poor absorption. If you don't absorb nutrients properly, that's going to result in some severe sickness. So again, ask yourself, hey, do we have one cell layer thick, or do we have more than one cell layer thick? And then is it flat, irregular shaped? Is it cube shaped, or are they column shaped? Okay. If they're squamous and they're irregular, remember they're good for diffusion, like in the lungs. When we have the cuboidal, we said we're going to find them in the proximal or distal convoluted tubules, which are found in the kidneys, good for secretion and absorption. And then the columnar cells we'll find also in the respiratory tract and the gastrointestinal tract. The pseudo stratified, remember, this is when we see the nuclei at multiple levels, they vary, and it has this false appearance of being stratified because the nuclei are at different levels. And often where we have pseudo-stratified, we're going to call it pseudo-stratified ciliated columnar epithelium. And transitional, this is a type of stratified squamous. We're going to find it in the urinary tract. So whether it's the urinary bladder, the ureters, or even parts of the urethra, these cells can be flattened when the organ is full, or it can be rounded when the organ is relaxed and empty. Now, where we're going to find some of these, well, simple squamous, which is one cell layer thick at the apical surface, this is great for diffusion, right? So we can have oxygen going this way, CO2 going the other way. We're going to find them in the alveoli of the lungs, which is really the air sacs of the lungs. The simple cuboidal epithelium, we find them in the kidney, where we have the proximal convoluted tubules and the distal convoluted tubules. Again, this is the tubule, right? Looks like a hose. And look, these cells are one cell layer thick. They're closely packed together. They're square, so they're cuboidal, and they're simple. Simple columnar epithelium, these are pretty much at one level, those nuclei. They're going to be found in the digestive tract. Whoop, let me go back here. In the intestines, the digestive tract. Pseudo stratified ciliated columnar epithelium, that's the respiratory tract. We could see those little hairs, those microscopic cilia over here. We could see that the nuclei are at multiple levels, but it's really one cell layer, but the nuclei are varied. So pseudo-stratified ciliated columnar, we'll find them in the respiratory tract. They often have goblet cells. Remember, those goblet cells are going to push out mucus to the external surface. Stratified squamous, look at these right here. Oh, this is all many layers of flat, irregular shaped cells. Yeah, you'll find them in the esophagus, you'll find it in the skin, the epidermis, right? So you want to have many layers of protection when you're eating food and it's going down the esophagus. If it gets scratched, it needs protection. You want to have many layers of protection on your skin. So if you scratch your skin with your nails, if it was only one cell layer, you'd be bleeding. So you have 25 to 30 layers of the stratified squamous epithelium protecting you. So the simple epithelium and stratified, look at categories there. There's simple squamous, simple cuboidal, simple columnar. For the pure stratified, same thing, stratified squamous, stratified cuboidal, stratified columnar. Then we have our pseudo stratified columnar, usually associated with cilia, which is found in the respiratory tract. And then transitional epithelium is the urinary tract, where it alters in their shape and size. Good place to take a break. When we come back, we'll get into connective tissue.